Good afternoon, everyone, and thank you for the invitation, and especially for the GRC for this specific invitation. So I would like to talk about guidelines for microfluidics and why guidelines and not standards, because most of the guidelines that we will produce in the coming years won't become standards because this is a long process. This is not mandatory to have standards for every guidelines that we will produce. Who will produce these guidelines? We have created 2 years ago Microfluidics Association. This is a non-profit organization which has been created following a European project, but this is an international association which is currently chaired by the NIST in the US, the National Institute for Standards and Tests, and which regroups the main key stakeholders in microfluidics, such as manufacturers like microfluidic chip shops or Microneat or Frigent, for example. Sorry. So I'm a board member of this Microfluidics Association and also the convener of the ISO working group and CN working group in charge of the standardization of the microfluidic devices. So how do we split the work between the Microfluidics Association and the ISO working group? Microfluidic Association works as a pre-normative group. So the idea is to build a large community of microfluidics I would say partners to develop the guidelines that we need in the coming years for the microfluidics domain. This is not focused on the MPS domain, but this is widely used for all the microfluidics development. Sorry. And once we have these guidelines, we can then transfer it, if needed, as a standard. So the Microfluidics Association is involved National Metrology Institute. And we have also a relationship with the FDA to build the next needs of standards. Okay. It has been identified by Monica that the ISO 10991 is in the scope of the organ-on-chips standard, and this standard is part of our group in ISO. So there is already an existing microfluidics definition which has been produced in 2009 in the previous version of the standard. And there is currently a new version of this definition, which is under revision. As introduced by Monica, there is the PSIS workshop, which has been held last year about the standardization of organ-on-chip. And during the microfluidics session, we have tried to build a roadmap, which is interconnected between the roadmap from the Microfluidics Association and the roadmap of the organ-on-chips. And what are the main topics identified during this session? Generic protocols for production and material characterization are essential. Testing and calibrations methods need to be standardized. Interfacing and various flavors of hybrid integration. Modularity is one of the promising aspects of microfluidics, and performance characterization standards for flow parameters and fluid leakage can facilitate interoperability. You can find all these details in the PSIS report that has been published by Monica and its team. So if you look on the chart on the left, you have the main topics placed on the chart depending on the importance and the feasibility. And I will address 3 points today. The first one, the terminology standards. We have to know what we are talking about before working on it. The second part, modularity and hybrid integration. That means interoperability between chips and the system. And the third part regroups a lot of things around the protocols and the catalogs of components. I will skip the materials thing because this is a challenging topic to find a consensus about the material to use in microfluidics, PDMS or not PDMS, etc. And as you can see, this is not the most feasible one, but maybe the most important one. We will see in the coming MPS World Summit if we have more advance in this topic. So first, the terminology standards. As described in the previous, in the first slide, there is a 10991 microfluidic definition. So this is a full standard with regrouping a lot of definitions with all the components in microfluidic, all the functions that you can have. We are currently working on the final revision. So this is a DIS version draft international standard, which is under ballot. That means that the countries are currently voting for this standard or not. And then we will publish the final standard by the end of this year. This will regroup a few categories: general terms relevant to microfluidics, general terms in microfluidics, flow-related terms, interfacing, and modularity. All these definitions could be used in the future work. About modularity and hybrid integration. So with the first standard really dedicated to microfluidics is 22916, which has been published earlier this year in January, which is related to interoperability requirements for dimensions, connections, and initial device classification. So this is a work started a few years ago through a European project, MF Manufacturing, and then published first in an international workshop agreement hosted by the BSI in London in 2016, and then pushed into an official standard published early this year. So the main part of this standard is to define the chip dimensions and the pitch position, and also application classes of the different components. I will just give you a brief overview of this standard. You can, of course, download this one and use it for your specific device. First of all, we have to define. Okay, yes. This is one of the drawing of the standard. You have the X and Y axis. No matter if this is top or side connection, you have to define a reference point, and then with the X and Y axis, you can have different inlet and outlets with a specific distance between the first port and the reference point, and also a pitch, which is a multiple of 1.5 millimeter. We prefer 3 millimeter for microfluidic components in polymer to avoid any leakage, but this is on your preference. Why did we choose a 1.5 millimeter pitch? This is because with this multiple, you can have the 3 millimeter pitch, the 4.5 millimeter pitch, which is compatible with the mini lure, and the 9 millimeter pitch, which is compatible with the micro tighter plate. So the idea is to have a broader compatibility with all the formats that we can find in microfluidics. It's more or less like the USB connector. You have different versions, but with the same objective at the end. We also have defined, so officially, microfluidics has official standardized format. The first one, of course, this is a microplate format because this is the main used format in microfluidics, especially in MPS, as we can see on the different booths. Also, the microscope slide is one of the format. I have indicated here compatibility with microplate format and with microscope slide because there are already existing standards for this specific format, but we have to adapt it to microfluidics. We have also defined an intermediate format for the credit card format if we want to use a size in between these two specific format, and also the microfluidic building block, which has been introduced by Andres a few minutes ago. The building block is a multiple of 15 millimeter for the sides, and this is a multiple, so you can have different versions. I illustrate here the interoperability that you can have with the thanks to the connectors. Because if you want to have interoperability with the system, you have to find the good connectors for that. So I illustrate with a few catalog components that you can find. For example, the micronit and the dolomite chip connectors, have a 3 millimeter pitch. They have been designed in compatibility with the standards, and especially for the micronit one, because it was not compatible initially with this pitch of 3 millimeter. So you can use the one that you want to connect your chip. And the third one, this is the row of 4 connectors provided by microfluidics chip shop, and this is with a 4.5 millimeter pitch, so compatible with the mini-lure chips. Also introduced by Andres, the fluidic circuit board. So if you want to have a modular and hybrid integration of your system, you can develop a fluidic circuit board with more or less three dimensions to place your different building blocks on it. We also provide clamping solutions. This one has been designed by Micronit, but as you have seen in the Andres presentation, there is also different alternatives. And I illustrate it with this top platform presented earlier. This top platform has been the first example published after the MF manufacturing project, because Twente was involved in this project. I used this standard to demonstrate the ability of the standards, of the guidelines, because it was not a standard at this period, including the microfluidic building block. Oops, sorry, this one. The microfluidic building blocks, which are clamped to the FCB here with a microplate format. You can see the different clamping solutions, and you can interchange all the building blocks as you wish. Other companies are introducing some modularity and some standards, and I have just an illustration of one of the company which are the booth today. This is a Netri with its Neobento solution. They were not involved in the MF manufacturing project. They just developed their own standard, but which fits with the 22916 standard. So the main advantage is this is a 96 well microplate format. There is a modular approach to experimentation, so you can easily change the different building blocks on the microplate. You can test it on the booth. And this is compatible with high throughput screening and with confocal imaging. So we can see that different companies are today developing the modules compatible with the future standard, with the existing standard, sorry. So you can also publish, if you are fully compliant, that you are ISO 22916 compliant. So the next part, which is ongoing work, because to work on this testing protocol, we first had to work on interoperability. Now we are able to connect with a good reliability the chips. We can work on the testing protocols. And the first part, which has been discussed with the FDA, this is the leakage testing. This is maybe the main point, the main concern for them. They want to protect the operator or to protect the patient from leakage. So the successful development and widespread use of microfluidic technologies are greatly dependent on the community's success in establishing widely supported test protocols. And one such area that requires consensus guidelines is leakage testing. This is an extract of a review that has been sent under peer review yesterday, which has been built with the FDA and the Microfluidics Association and should be available quite soon. We have also published today on ResearchGate a white paper about the protocols for leakage testing. So this is more review about the leakage testing, and now we are working on the future protocols to be standardized. And to do that, I introduced in the beginning that we work with National Metrology Institute. This is very important to have them on board because we are able to provide a roadmap to explain what we want in the coming years, but we are not able to develop very precise protocols. That's why we have been funded with Euramet, the European Association of Metrology, and Empire participating project. The project establishing metrology standards in microfluidic devices. So this project aims to contribute to the development of globally accepted standards for microfluidics. So we try to develop measurement protocols and guidelines and the dissemination of metrology standards through the different normative committees, especially the one of microfluidic devices. So you can see that there is interconnection between all the main stakeholders, the FDA, so the regulatory, the Metrology Institute, the Microfluidic Association, and the Normative Association. And another ongoing work, this is AWI, so that means approved work item. So it has been voted last year. This is a work item about microfluidic pumps, the symbols and performance communication. The idea is to, through this first example, so it won't be a standard, it will be a technical specification, but the idea is to do a first tentative to build microfluidic datasheets. Because we, as end user, we want to be able to buy a microfluidic component with the associated datasheet. And for that, we try to do this exercise with the microfluidic pumps. So as a conclusion, microfluidic community is addressing the main standardization topics for OoCs. You can join this microfluidic association if you want to contribute to this roadmap and to help to the white papers and different contributions. And standards are a tool to improve quality, to ease integration, and to accelerate innovation. Sometimes we think that if we have to follow standards in research, it will block the innovation, but it's not the main idea. The main idea is to provide guidelines to save time, to have reliable components and connections. And my last slide, I would like to acknowledge, of course, MFA board members and other members, and also the ISO working group experts, which is an international group. And also to mention that Grenoble is hosting the EUROX yearly conference, the 4th and the 5th of July. So you can join us if you want to discover the French Alps. Thank you very much.